Pages 22 and 23 detail the work done by Sir Alan Hodgkin and Sir Andrew Huxley. Both scientists worked in England and conducted experiments focused on isolating a single neuron from a squid. The men chose a squid because squids have bigger neurons than mammals, which made it easier to isolate. The experiments conducted tested the travel of nerve impulses along the axon. They discovered that neural firing was electrochemical. Based on this finding, the men learned voltage increases along the axon are caused by changes in the membrane of a neuron. This change permits positively charged sodium ions to rush quickly into the axon from the outside. They referred to this phenomenon as action potential. The membrane then again quickly changes as it pushes out positively charged potassium ions. These ions are pushed out to restore the membrane to its initial resting voltage. Then on the bottom of page 24, the book starts discussing neuroimaging. Neuroimaging is defined as the set of methods that generate images of the structure and/or function of the brain. Many advancements have occurred to make neuroimaging even better than it was in the past and more prominent in use. The book then decides to talk about electroencephalography, also known as EEG, which measures electrical activity through multiple electrodes placed on the scalp. EEG cannot pinpoint individual neurons. It is only primarily used to roughly localize neuron populations, but it can do that very accurately. Just one result isn't always the most accurate, so scientists have turned to using event-related potential. Event-related potential, also known as ERP, is the average electrical activity from a group of neurons based on many EEG readings. ERPs are also measured in microvolts. On page 26, the book then discusses another method called magnetoencephalography, also known as MEG. MEG measures magnetic activity across populations of neurons in the brain. MEG has better spatial resolution than EEG. However, MEGs are much more expensive because they use superconducting quantum interference devices. The book then briefly discusses X-rays in regards to computed tomography. Computed tomography is an imaging technology that uses X-rays to create images of slices through volumes over material. X-ray cannot see the brain's tissues. However, CT can. Then, right after talking about CT, the book goes into magnetic resonance imaging, also known as MRI. MRI is an imaging technique that uses the responses of atoms to strong magnetic fields to form images of structures in the brain. This method can also be adapted to focus on brain activity instead of just brain imaging. MRI focuses on pulsating magnetic fields. Then the text continues with, on page 28, by talking about functional magnetic resonance imaging, also known as fMRI, which shows the activity of the living brain. The brain is a tissue and therefore it needs oxygen. When it needs more oxygen, something called blood oxygen level dependent signal occurs, which is when the brain sends out a signal demanding more blood, therefore giving it more oxygen. These fMRI machines are slow and noisy and very expensive. However, they have been very influential in helping the study of the brain. The final method the book talks about is positron emission tomography, also known as PET. This is a technique where a safe radioactive material called a tracer is injected into the bloodstream. After this injection, a specialized camera detects gamma rays, and it is used to watch neural activity by watching an increase in metabolic activity. And then the book finishes off by talking about how most labs use the majority of these in their work to get a full, better reading. And on page 29, the final page of this chapter, is a summary of the most important ideas that occurred.